We're privileged to have a 30-minute talk that will be delivered by Professor Ashley Bush, one of the world's leading dementia researchers. After Ashley speaks, we'll then have reserved 5 to 10 minutes for question and answers. Please message your questions through the chat function as the lecture plays out. We hope to have enough time to answer a number of these, but we do ask that you keep your questions short to help us get through as many as we can. If we don't get to your questions today, we'll endeavour to respond to you directly over the next week. Professor Ashley Bush is the Director of the Melbourne Dementia Research Centre at the Florey Institute of Neuroscience and Mental Health, an NHMRC Research Fellow and a Professor of Neuroscience and Psychiatry. He's Co-Director of Biomarker Development for the Australian Imaging, Biomarkers and Lifestyle Study of Ageing, and holds staff appointments in psychiatry and radiology at Massachusetts General Hospital, USA, and as a Professor of Neuroscience and Psychiatry at the University of Melbourne. He's a scientific founder of multiple biotechnology companies such as Cogstate Limited and Collaborative Medicinal Development. And if that wasn't enough, he's also been rated as one of the world's most influential scientific minds by Clarivate Analytics. So without any further ado, I will now hand over to Professor Ashley Bush. Over to you, Ashley.
If you have any trouble hearing me, please write a note on the question and answer board, and I'll try to fix the problem as we go along. I'm sorry about the technical problems that we started with. So here's the title of what we're going to talk about today: Alzheimer's disease, aging and the search for the cure. Now, this obviously can't be covered in any particular depth in 30 minutes. But what I'm hoping to do is to just give you a little taste of what's happening currently and focus down on the work that's being done on the Florey, where we're doing some exciting stuff that I'd like to tell you about. And we can cover any of your general questions, as Steve says, towards the end. So Alzheimer's disease, a very common diagnosis nowadays. This is a very nice illustration that I like to use about what we call the natural history of Alzheimer's disease, that being the how the disease normally progresses in terms of its symptoms. So this is a series of self-portraits that were performed by an artist called William Utenmolen, and this work was published a couple of decades ago now by Martin Rosser. And these self-portraits show the artist drawing himself from the age of 60 when he was given the diagnosis of Alzheimer's disease over the subsequent 6 years, and then he died the year after this last portrait. And you can see the slow deterioration that occurs and the disassembly of his abilities to organize and to portray accurately what reality is. Although, nevertheless, throughout, he does actually retain his artistic talent and is able to, even with advanced Alzheimer's disease, make a drawing which we would appreciate as being quite substantial and interesting. So Alzheimer's disease was described first by Alois Alzheimer, a German psychiatrist, in 1906. Here's a picture of him. And this is his first patient, Augusta D. And she was put into his mental hospital, and she died in her mid-50s with a severe psychotic disorder. He dissected her brain and found these telltale pathology that I'll show you in the coming slides. And Alzheimer's disease is described as a neurodegenerative disease. That is, that it's a damage to the brain tissue and the nervous tissue, which slowly gets worse and worse with the brain cells, the neurons dying off. And it's classically characterized by short-term memory loss. This is the most common and earliest symptom. But it's a gradual deterioration. So you don't develop a diagnosis of Alzheimer's disease overnight. The symptoms of memory impairment get worse and worse and worse over a few years. and then eventually cause sufficient incapacity, so it brings you to the attention of a physician, and the physician will eventually make the diagnosis. But there can also be changes to personality and behavior because of damage through other parts of the brain. And as the disease gets very severe, it can lead to a loss of bodily functions, and ultimately death occurs on average 7 to 10 years from the diagnosis. So dementia is any condition that causes a loss of cognitive function. Specifically, we mean memory and reasoning, which are the computer functions of the brain. So generally speaking, dementias don't cause motor abnormalities. Those are difficulties with movement, such as Parkinson's disease, although it's quite possible to have the two conditions simultaneously. But for dementia, we refer to loss of brain function, cognitive function. Alzheimer's disease is the most common cause of dementia, representing about 70% of all diagnoses of dementia. And this pie chart shows you how we slice up various diagnoses of dementia, Alzheimer's disease representing the biggest slice of the pie. The other causes of dementia include vascular dementia, which is the second most common cause. And then, of course, there are some disorders which there's a mixture of both vascular and Alzheimer's disease. Other causes of dementia include frontotemporal dementia, Lewy body dementia, and even Parkinson's disease itself eventually becomes complicated with dementia if it's advanced enough. And the classic thing that is seen in Alzheimer's disease is that the brain shrinks. This is a picture of a normal healthy brain, which has the size of a large cauliflower and weighs about 1.5, 1.6 kilograms. And then in Alzheimer's disease, you get this loss of the meat of the brain, and you see enlarged spaces between the folds of the brain's tissue. This is called atrophy. Now, what Alzheimer himself saw down the microscope in 1906 were these two phenomena, the amyloid plaques and the neurofibrillary tangles. Now, the plaques here are 3 plaques which are stained brown. Here you see in the other slide the neurofibrillary tangles, and work from the mid-1980s onwards, including the tremendous work of Colin Masters, who is at the Florey Institute, defined the main proteins that are accumulating here, because the proteins are the working molecules of all biology. And if we get the chemical composition of this type of lesion, we can perhaps understand more about what the cause of the disease is. So the main component of the amyloid plaque is a molecule that was sequenced by Colin Masters and others in the mid-80s, and it's called A-beta. That's its biochemical symbol. And the main protein that's accumulating in these dark neurofibrillary tangles, which are filling up the brain cells, is called tau. These have been the main 2 proteins of interest in Alzheimer's disease. And we've tried for many years to figure out why these proteins accumulate in these sort of patterns in the brain, and whether this is the cause of the disease, and whether if we stop this accumulation, that would be a treatment for Alzheimer's disease. And to cut a long story short, 30 or more years of research into this has not been able to deliver a simple answer. We're still trying to understand why this happens and how it's linked to the disease, and whether you can have a treatment that targets this that would be effective in treating or stopping Alzheimer's disease. We've come a long way over the last 30 years in being able to make the diagnosis accurately. So Alzheimer's disease is no longer considered a normal consequence of aging. It's an unusual complication of aging, which affects about 5% of people over the age of 60. And we developed scans for these amyloid plaques that Alzheimer first denoted. And this is a typical amyloid scan, where you see a normal person on the left and a person with advanced Alzheimer's disease on the right. Everyone with Alzheimer's disease has got amyloid within their brain. And a perfectly healthy brain does not have amyloid within it. This is considered to be the background signal. And this red stuff here is the signal that is considered to be the amyloid. But the problem is that there are quite a few people who've got amyloid within their brain, but they're actually cognitively normal. Are those people more likely to develop Alzheimer's disease? Yes, they are. Will they always develop Alzheimer's disease? No, they won't. So the presence of amyloid alone is not enough to predict that a person is going to develop Alzheimer's disease. And this amyloid builds up over a period of 15 years on average before a person comes down with a diagnosis of Alzheimer's dementia. So the amyloid has been a very important part of the research into Alzheimer's disease over the last 30 years. And particularly in the last 20 years, there have been a number of attempts to try to clear out the amyloid from the brain with various drugs. And so many attempts have been achieved right now that I want to list for you just the late phase 3 clinical trial results from these amyloid trials, just to make a point. Now, there were many reasons to think that amyloid was going to be the appropriate target for therapy in Alzheimer's disease. But sometimes being confident about a prediction doesn't guarantee in science that you're going to be able to achieve a result in the clinic. And that's part of the mission of the medical research, is that we have to be careful in assuming that we know the cause of diseases until we've actually got a drug that works. So in medicine, you can have the best scientific theory, but if you can't make a treatment out of it, or you can't make a drug out of it, then your theory is not yet valid. And I'm afraid that this is where we are with the amyloid story, that there have been 34 phase 3 clinical trials that have not reported benefit, and in some cases have actually made the disease worse. And the estimate cost of all of these trials has been astronomical, $18 billion. Most recently, the one that's in the press is aducanumab. There is some evidence that that aducanumab may have a benefit in a clinical trial, which is done by Biogen, and it has been taken to the FDA. Even if aducanumab becomes registered, it will still only have a marginal benefit, if at all, for Alzheimer's disease treatment, which is being debated currently. So the bottom line is that amyloid itself does not appear to be a very conspicuous target for treatment in Alzheimer's disease, in the sense that it's not likely that a lot of benefit would come from clearing the amyloid out in the brain. It seems to be like spots on your skin in measles, that the spots are not the cause of the measles, and the spots in the brain, the amyloid, is not the cause of Alzheimer's disease. It might be normal, but it's not the actual thing that's killing off the brain cells. And here's a quote which summarizes where we are with the amyloid hypothesis. So we've come to the conclusion that the plaques are not the cause of senile dementia, but only an accompanying feature. And this highly contemporary analysis comes from no less than Alois Alzheimer himself. After 5 years of working on this, he came to the conclusion that the amyloid is not likely to be the actual cause of the toxicity that kills off the brain cells in Alzheimer's disease. And in terms of modern editorials on this, there's been a call to rethink Alzheimer's disease. Here's a title page from the New England Journal of Medicine, the most prestigious medical journal, recently saying that we're probably nearing the end of the amyloid hypothesis rope, in which case eventually we need to loosen our grip of it and look at other things. The field is in need of innovative ideas. Now, this was a journey that had to be taken. So now we're in an era where other approaches to thinking about Alzheimer's disease are emerging. And we have to come up with something, because Alzheimer's disease is a very big problem. It's estimated there'll be 135 million people affected by it by the year 2050. 1 in 3 people over the age of 85 have dementia, and most of those have got Alzheimer's disease. And it's going to be an expensive disorder to treat because there's, as I've mentioned, there's no cure. There's some palliative treatment. There are drugs that can be taken that boost function a bit, but not drugs that actually slow down the death of the brain. Financial cost to Australia is very severe, and about 3% of GDP will be lost to Alzheimer's disease care by 2050. Currently, it's a diagnosis which actually can be made not just from autopsy or brain biopsy, but also from amyloid scan in tandem with the clinical presentation. We are constantly working on diagnostic and predictive tests. So this is something that we hope we're going to have valid over the next 10 or so years. So there's better work being done towards making a prediction of a person with Alzheimer's disease. But right now, there's no scan and no blood test that you can have that would actually predict that you're coming down or will come down with Alzheimer's disease. That needs to happen. So the diagnosis is generally by clinical assessment, excluding other causes of dementia. A number of tests are done to make sure that a person doesn't have another contributing cause to their loss of function. There, I've managed to turn on my video. So if you want to see me, I'm here. Thanks for being patient with all of this. The treatment, as I mentioned, is largely on the basis of symptoms, and the main drugs that are used are Aricept and rivastigmine. There are some other ones such as Bixor and Namenda, and there are other things to treat in Alzheimer's disease, such as treating the complications of depression or epilepsy. And over the years, there have been a number of over-the-counter drugs that have been looked at, and of course there are a lot of experimental treatments that are being examined outside of the amyloid hypothesis. So I want to tell you now a bit about our research on trying to find a treatment for Alzheimer's disease. Now, with this disappointment in the amyloid hypothesis, we went back to the very beginning and we said, well, Alzheimer's disease has as its major risk factor aging. And that's not a coincidence. There's something about the chemistry of the brain that occurs that sets up conditions for the amyloid and the tau to accumulate within the brain. And we know that obviously the brain undergoes chemical changes with aging, and we want to see which one of those chemical changes sets up the conditions that might lead to neurodegeneration. So when we stood right back from this picture, we asked, well, what about the chemistry of aging might set up these conditions for Alzheimer's disease? And we went back to evolutionary theory, that life on Earth commenced 3 billion years ago with the emergence of iron in the Earth's crust. And at that point, this iron was used for self-replicating molecules to generate the organisms that subsequently evolved into us. So life on Earth depends upon iron. And over the history of biology, the biggest problem for developing organisms was the limitation of finding iron in nutrient sources, that there is not enough iron to go around. And even microorganisms fight with us for that iron. And because iron is limiting for development and reproduction, no pathway ever evolved for the excretion of iron, which is very interesting. The only way you can lose excess iron is to either bleed or to shed your gastroepithelial cells. And these are very inefficient or undesirable ways of losing iron. And what we knew as a clue from the early work in Alzheimer's disease is that there seems to be a buildup of iron within the brain. And in any tissue where this occurs, it can induce a type of oxidation which is equivalent to rusting. So a kind of tissue rusting, which particularly affects the lipids, the fats within the cells. And there's a lot of fat in the brain. 20% of the brain is cholesterol. This biological phenomenon of iron rising needlessly with aging has been described in a lot of experimental systems that you wouldn't think immediately have got anything to do with Alzheimer's disease. For example, this is one of the earliest reports I could find in 1985, measuring the amount of iron in flies as they age. A fly lives to about 60 days of age. On the y-axis here is the level of iron in the flies. As the flies age, the iron accumulates and accumulates. Now, a fly doesn't need all of this iron. It just is a product of its biology. It just will mop up the iron as it goes along. Now, we thought this was very interesting because this unnecessary accumulation of iron could be something that could set up conditions of oxidation, which we know occur in Alzheimer's disease. And indeed, when we measured iron in mice, we found that it was elevated in the brains of these mice as they age past their reproductive life. So a mouse stops reproducing around about 6 months of age, but nevertheless, the levels of iron as the animal lives longer past its reproductive life continue to rise in the brain. And this phenomenon found itself to be reproduced in other biologies, in mammalian systems and then in humans. So mammals, all humans, all primates, a whole bunch of papers have now appeared that shows the same thing, that as you age, the concentration of iron in the brain goes up for no good reason. So we decided at the Florey to study this in great depth and to determine whether or not this is truly responsible for hazard, for causing toxicity, or does the iron just get stored away and it just sits there? And we studied this animal here, the C. elegans. This is a 1 millimeter long worm that we can study in the lab, and a lot of aging research is done with this organism, the Cynorhabditis elegans. And here is a dissected worm cut into 3, its head, the middle, and the tail, and it's stained for iron. And you'll see quite vividly that as the animal ages to 12 days of age, and it lives to 20 days of age, but at 12 days of age, the animal is full of iron. The animal reproduces around about 4 days of age. So this is a post-reproductive life worm. And this is where the iron senselessly accumulates. And the trapping of the iron within the worm can be pharmacologically trapped with an iron trapping drug. And we have recently published, this is actually hot off the presses because it was accepted this week in a prestigious journal called eLife, that if we trap this iron in the body of the worm, that we can extend its lifespan. So I'm going to show you the data. This is the work from Gavin McCall and Nicole Jenkins in our lab. On the x-axis are the life days of the worm from 0 to 35 days. And on the y-axis is the percentage of animals that are alive in a colony of animals. And this, we studied about 1,000 animals in total all up. And here you see a typical survival curve of a colony of worms, that there's 100% stay alive till around about 6 days of age, and then they start dying off. And then by the age of 22, they're almost all gone. Now we see what happens when we treat these animals with this iron trapping drug. And we see this dramatic doubling of the median lifespan of the worm. So this excess iron that's accumulating in the post-reproductive life of the worm is hazardous. And we can extend lifespan this way. These worms are healthier and fitter with less iron in them. And this is an interesting principle which we think generalizes to all forms of aging and applies to the brain in Alzheimer's disease. And we know that it's relevant because of several layers of evidence that we've come up with. And this is typical of the kind of neurological research that has to be done when you're trying to figure out what causes a disease where the cause is actually not known. So we've studied post-mortem human brain tissue from people who've donated their brains. We've studied fluids such as the cerebrospinal fluid that coats the spinal column and is in contact with the brain. And we've studied MRI through special sequences that allow us to measure the content of iron within the brain. I want to show you a little bit of this results just to show you how this research marches forward. Alzheimer's disease is a slow process, And we use the measurement of amyloid as one of the index to tell us that at least the disease could potentially be present even if the person doesn't yet have symptoms. So we can measure the amount of amyloid within the brain over a lifespan, well, from adulthood until advanced age. So we have volunteers who've been coming in and having themselves scanned over periods that are now measured in decades. And we know that the amyloid takes about 15 years to accumulate before the symptoms of Alzheimer's disease come on. And I mentioned this earlier, that a person with a positive amyloid scan doesn't necessarily have Alzheimer's disease, that there's a big overlap. There are lots of people, HC stands for healthy control, healthy people. There are a lot of people who've got a positive amyloid scan that looks like this, but they actually have got no abnormalities in terms of their cognition. They haven't got Alzheimer's disease. And then there's a group in the middle, the mild cognitively impaired patients, who have got some memory problems but are not fully demented. And you can see the big overlap that occurs. This group here, amyloid positive, what's going on there? That's what we would like to know. We've recently published that what might be going on is that it's only when the iron levels go up in the brains of these people that the amyloid starts to become a problem. And here's some of the data from the paper that we published on this, in which we measured the iron levels in people who died with the amyloid in their brains. And we determined how much iron was in those brains. And in this analysis, we've categorized them as either being low, medium, or high. This group, we had neuropsychological performance data for the 12 years prior to death. So this is a volunteer group who came in on an annual basis and had neuropsych testing about their cognitive performance done for up to 12 years before they died. And we received their brains and we measured the amyloid and we measured the iron levels. And what it shows is that the amyloid alone is not enough, that in tandem with the high iron level, the cognitive deterioration, which is measured on the y-axis, is accelerated. So the loss of cognition is accelerated in people who have both high iron and amyloid. So this is an additional risk factor in terms of whatever it is that causes the degeneration to occur in Alzheimer's disease. We see something similar when we measure iron in the cerebrospinal fluid. And here we have the baseline levels of ferritin on the y-axis, and the let me explain this again. This is this is like the worm study, a survival cohort of people with a mild cognitive impairment, and we're seeing how long does it take them to convert to Alzheimer's disease. And at the baseline, we measure their iron levels represented by ferritin in the CSF. And we've divided the cohort of 144 people into quintiles, 20% intervals from the lowest levels of iron to the highest levels of iron. And it's those with the highest levels of iron who convert to Alzheimer's disease fastest, about 2 years faster. So elevated iron in this cohort induces an earlier onset of Alzheimer's disease. This is some work that we published over the last few years. Finally, MRI, and here is the imaging machine, which gives us an image of the brain, which is measuring the iron content in the tissue. It's a different way of using this MRI technology. And it's very useful because it allows us to basically do the same as either directly measuring the tissue from people who died or taking a lumbar puncture, but in this case, it's non-invasive and we can actually see the iron in the brain itself. And using this, we get the same type of result as we did with the CSF analysis. The people with the higher iron level deteriorate faster over a period of 6 years. Okay, so I'm going to finish up here. I really just gave you a little taste of what's going on with iron. But I like to show people what's involved with trying to find the money to do this research, because the university doesn't pay us to do research. This research is funded by ourselves through raising money from grant sources. And there are a couple of governmental grant sources where we apply for money. What people might not realize is how difficult it is to raise this money. What you're looking at here is a grant application. It runs at about 106 pages long. The probability of funding is roughly 12%. Now, anything that we've done as an experiment that we've published here has been done because of money being raised through this mechanism. So it's very laborious, and you have to be very patient and very determined to be able to succeed to raise the kind of money that's required. These kind of studies are very expensive, but obviously this pioneering work has to be done, or else we're never going to be able to get interventions for these incurable disorders. Thanks to a grant from the NHMRC, we are now testing whether lowering brain iron using a drug called deferoprone can delay dementia in people who have already been diagnosed with Alzheimer's disease. This is the 3D study. We're still enrolling it. The National Health and Medical Research Council awarded us $2.5 million to do this study, and we started the trial in 2017. If you know someone who has got Alzheimer's disease and is still living at home, you can send us an email to 3d@florey.edu.au, and we'll see whether the person might be eligible to be enrolled in the study. So with that, I'd like to thank you for your attention, and hopefully we'll be able to avoid our iron accumulation in our brains long enough to be able to celebrate our centenaries. Thank you.
Thank you, Ashley, for that wonderful talk. I hope if everyone can please join me in giving Ashley a big virtual clap. I don't know how we do that, but well done. And it's great to hear the latest in the thinking, and hopefully one day, Ashley, we'll be talking about your successful trials and how we start to turn the corner on this terrible disease. There have been a few questions that have come in for you, Ashley, and I'll just read them aloud. If you could please answer them after I read them. One came from Cindy that read, I just read about Recode by Dr. Dale Bredesen. Have you heard of this protocol? And if so, is it worth considering?
Yes, I know Dale Bredesen, and I know of his work, and he's actually a really good scientist, and he's put together the evidence that's available about approaches that are not prescription drug approaches to potentially help with Alzheimer's disease. He comes from the thinking that there are a lot of potent over-the-counter chemicals that are available that will never be studied in the kind of large-scale clinical trials that we need in order to create a prescription medicine, because the money that's needed for that can only come from big drug companies. We can do research on repurposing, like I showed you the deferoprone study. This is a repurposed drug which takes only $2.5 million to show whether or not it has a benefit. Now, Dale Bredesen has said that, usually it takes hundreds of millions of dollars to determine whether a drug is worthy of being prescribed as a prescription. So he said, well, it's not going to happen for the majority of things out there like fish oil and selenium and, aspirin, let's say. There are lots of potential drugs out there or products which might be beneficial, but we're not going to know for certain until someone tests it out in very large-scale clinical trials. He's put a compendium together of the best guesses of what might be useful. And it is actually a scientifically quite valid analysis of the possibilities. So I like the work. I refer to it myself in terms of the treatment of my patients with Alzheimer's disease. It involves taking quite a few supplements, which can, in their own right, be expensive to buy all of these things. But if you're very motivated and you're willing to embark upon that, I think what his advice is pretty good, actually.
Thanks for that, Ashley. Roger Henderson asks, it seems to me that tau and A-beta are the end result of a disease process occurring silently in the background. The brain is a very high energy consuming organ. Long-term small shortfalls in energy supply is surely a trigger for a number of defects in protein structure, especially kinase, phosphatase balance, etc. So I guess asking about how energy fluctuations and demands may contribute to the disorder.
Yeah, almost certainly energy loss is going to be contributing to aging and to the disorder. All the pathways that you mentioned are indeed impacted by energy loss with aging. And that's an area of important research in its own right, how to maintain energy levels with advanced age chemically, how to get the fuel to keep the cells metabolizing efficiently. That's another very big and very worthy subject matter in its own right, which applies, of course, to other things apart from dementia. So, yeah, very valid point.
All right, one last question. I realize that we're running at the top of the meeting. This comes from Deb, and is the disease a relatively modern one, 150 years or so, or has it been with us forever?
It's been with us forever. It's just that people haven't reached the age of risk on average as much as we do nowadays. So medicine's been very successful in getting us to live longer, and we survive diseases which commonly used to kill us quite frequently, like heart disease and cancer. And then we live to the age of risk where these neurodegenerative disorders such as Alzheimer's disease become more prevalent. So these diseases have been there forever. We're just seeing them more commonly now because we live on average longer.
One of the unexpected side effects of improving medicine.
Yeah, that's right.
So I might wrap up now. Thank you again, Ashley, for a brilliant lecture. And I'd also like to thank all of you who joined us today. We appreciate your interest in the Florey's work. We will be sharing a video of today's talk on our website and social media channels. So please keep an eye out for this in the coming weeks. And if you did enjoy our talk today, you may be interested in registering for our next online lecture on the 19th of August, which will feature our latest work in sleep disorders and the brain. Finally, please don't forget to email or call our fundraising team if you have any questions or would like to give to our research. Thank you again for joining us, and I hope you and your family are staying safe and healthy. I look forward to keeping you informed about the Florey's research. Goodbye, everybody.
Bye-bye.